Hi there and welcome to this A-level revision video by Mr Phillips, and today we're looking at polymerase chain reaction where we're making multiple copies of DNA artificially. This is the second of the two methods of how we can make multiple copies of DNA. So last time we looked at in vivo, which is using bacterial inserted into the plasmid and then inserting that into the living cell. This time we're using amplifying it using a PCR, which is in vitro. At the end of the video we'll compare the two and the advantages and disadvantages, which is a common example. DNA reaction is an artificial method of DNA replication that can make large copies from tiny samples of DNA. Vital in forensics, where the evidence might be extremely small, it might be ancient DNA, it might be modern. The DNA can be amplified enough to do genetic processing. The whole process relies on some key facts about DNA, that it's made of anti-parallel backbones, it's made of 5' to 3' end strands, and the new strand grows only from the 3' end because the RNA polymerase, or DNA polymerase I should say, attaches to the 5' end of the template strand. There is also complementary base pairing occurring, as you should know, and all those factors mean that we can make. You need to be aware of the method of DNA replication and understand that, because obviously it involves a lot of enzymes and there are some stages you could be asked for differences between the two. Is it different from DNA replication? Well, this only copies a small sequence of DNA, a few hundred base pairs in length, so you may then need to add other processes to make more longer strands from what you create. It is using the addition of a primer. Now, a primer is a short sequence of nucleotides that allow the end of the DNA strands of the two strands to be double-stranded. So we'll start off with two single strands of DNA. Imagine these are bases I'm adding on now. In order for DNA polymerase to attach, each strand needs to have a double-stranded bit at the start. So a primer is this double-stranded bit at the start that I've drawn there, which will allow then the DNA polymerase to attach to continue the process for us. So a primer is a short sequence of nucleotides with complementary to the DNA at the end of the strands of both fragments, allows DNA polymerase to attach to start the process. And the whole process relies on a cycle of heating and cooling used to separate and bind DNA strands. And basically in the natural process we know DNA helicase is used, but so we're only using one enzyme, polymerase here, that is removed by using temperature as a control. The enzyme we use is DNA polymerase, as I said a few minutes ago, but it's a type of DNA polymerase called Taq DNA polymerase, so that's TAQ. Okay, and Taq DNA polymerase is DNA polymerase from thermophilic bacteria in volcanoes, which means it's got a optimum temperature of much higher than. That controls all this, which is controlled using varying temperatures, is called a thermocycler. What we need to know about polymerase chain reaction. So we start off with our double-stranded sample of DNA, which might be a small sample of up to 100 base pairs in length. We then use heat to separate that. So at 95 degrees C, the bases will break, the hydrogen bonds will break between the bases, and we'll end up with our two separate strands. We then add the primers and reduce the temperature to 55 degrees C, where DNA exposed bases naturally anneal. So the primers, which are complementary to the initial strand endings, will then attach. We then raise the temperature to 72 degrees and add 3 nucleotides in, and that then allows DNA polymerase from the Taq bacteria to anneal the exposed bases to create two new strands of DNA. We then do the process again and again and again, and the numbers will go up exponentially. In vitro and in vivo, there are lots of comparisons that are very useful to know. So if we have a look in vitro, the PCR can copy DNA that's been partially broken down, useful in forensic scientists, but partially broken down will not be used in bacteria. PCR is very sensitive, so minute amounts of bacteria can be copied, whereas less sensitive and we need larger amounts in vivo, so it's less useful in forensics. DNA embedded in other materials can be copied in PCR, whereas it has to be isolated in vivo. The clones are produced in a solution, and they cannot be used to directly manufacture it in PCR, but in vivo we make it inside the bacterial cell or inside the goat's mammary gland cells, and we can make the protein as a result. The DNA polymerase will only copy a gene if it is marked at the end with primers, and if we don't know the base sequence at the end, we cannot use PCR. But once it's incorporated into the host DNA in vivo technique, we can copy any and make any DNA again and again. PCR becomes unreliable if it gets too long, whereas in methods it reliably copies up big genes in vivo, and in vitro lacks any error correcting mechanisms, whereas cells will often correct any errors as they go around, so it'll be less copy. Or PCR, the benefits are it is rapid and it will use tiny amounts of DNA and you can still get lots out. It does not require living cells. But the issues are if you have a contaminant piece of DNA, so some DNA you don't require, that will also be replicated, and up to 20% of the DNA made will contain errors in from the process. In gene therapy, because we're trying to introduce it into a organism, there is no contamination risk because only the DNA is treated with restriction. Endonucleases will be uptaken. It is more accurate because there's rarely any DNA errors because mutations are so rare. You can use specific genes rather than a whole DNA sample, and there is a high production. So once making DNA, so we can make once it's into the bacteria, it's very rapid to produce huge quantities.